Thank you for watching my presentation on molecular approaches for eelgrass conservation. My name is Brian and I am a graduate student at the University of Washington. In this talk, I will explore the evidence for adaptation and plasticity in the phenotypic expression of life history traits in a key seagrass species. Zostera marina, commonly known as eelgrass, is a flowering plant widely distributed throughout the Northern Hemisphere and along the west coast of North America, where it thrives in a wide range of environmental conditions and consequently exhibits extensive phenotypic diversity in numerous traits. But an intriguing life history characteristic of eelgrass is its capability to reproduce asexually via cloning, which is also associated with perennial life cycles, and sexually through seeds, which is associated with annual life cycles. The preferential allocation to either mode of reproduction in specific conditions remains unanswered, but foundational work has provided evidence suggesting that the expression of these traits is influenced by complex relationships between the environment and genotypes. My questions are then, is phenotypic expression of life history traits in Zostera marina driven by phenotypic plasticity or local adaptation? And is there evidence for genotype by environment interactions? To address these questions, we conducted reciprocal transplants, experiments used to study the extent of adaptation in the expression of quantitative traits. We had two sources, perennial and annual, here on colored blue and red. 4 outplant locations, an annual site where shoots predominantly flower, and 3 perennial sites where asexual reproduction is prevalent. Each site contained 5 plants from each source, and we translocated 25 individuals into each plot, for a total of 1,000 transplants. We recorded trait data on cloning, flowering, and shoot length, and analyzed this information using mixed effect models to evaluate the extent to which the expression of these traits is influenced by source or genetic effects, outplant sites or environmental effects, and their interactions, which is used to test the relationship between environment and genotypes. We also collected tissue for genetic analysis, but the molecular work is still underway. We do have preliminary ecological results. Clonal investment was measured by the number of branches produced on average by each plot. Perennial plants invest more into clonal reproduction, and as observed, this pattern was retained regardless of outplant site, suggesting an adaptive association between perennial shoots and enhanced branching. However, the relative pattern of differential expression between sources also varied among outplant sites, indicating a genotype by environment interaction modulating asexual reproduction. Flowering effort shows a similar but inverse pattern. In this case, annual plants retained their life history strategies and preferentially flowered in all outplant sites, indicating the influence of a genetic component favoring flowering in annual source shoots. Interestingly, there were no significant associations between the allocation to flowering and outplant site and with the interaction between source and outplant site. These results show a significant genetic source differentiation driving the expression of sexual reproduction via flowering. And lastly, maximum shoot length, measured in centimeters on the y-axis, is also significantly influenced by the interaction between source and outplant site. As the graph shows, perennial source shoots outperform annual source ones only in some of the outplant sites, suggesting evidence for a genotype by environment response in determining the length of shoots. To summarize, there was evidence that life history traits were retained from the source population, suggesting genetic differentiation between annual and perennial plants. There was also evidence of significant genotype by environment interactions for cloning and shoot length, but not for flowering. Complementary high-throughput genetic work will further this research to understand the molecular mechanisms influencing the phenotypic expression of traits in Zostera marina. This study will help to clarify the long-standing debate concerning allocation to reproductive modes in eelgrass. But most importantly, we aim to inform the adequate selection of eelgrass transplant donors to improve coastal management and ecosystem restoration efforts. Thanks so much for your attention.